Welcome to Lecto. Today we are going to discuss different sources of the vitamin K, that is actually the fat soluble vitamin, and the functions of the vitamin K. So let's start. First of all, we will discuss the different sources of the vitamin K. First one is the cabbage, second is the kale, third one is the spinach, and fourth one is the egg yolk, and the last most important is the liver. So simply is that green leafy vegetables contains the vitamin K. Okay. So if we discuss the requirements, different requirements of the vitamin K. So in case of adult male, the requirement is 120 microgram per day. Okay. On the other hand, female required the 90 microgram per day. Another important source of the vitamin K is the bacteria, intestinal bacteria. Intestinal bacteria also produce the vitamin K, specifically K2 form of the vitamin K. Okay, so if we quickly revise the different forms of the vitamin K which we have discussed in the previous lecture. So first one is the phyloquinone, that is vitamin K, that is actually the plant source. Okay, Managunon, vitamin K2, that is actually the source of intestinal bacteria, actually the source of vitamin K2. Manadiones, that is the synthetic form, and the different vitamins K form that actually form during the reaction, that is the hydroquinone and the epoxide form. So here are basically the different forms of the vitamin K. If we discuss the functions of the vitamin K, okay, the most important functions of the vitamin K is in the post-translational modification. So if you see here, first of all, here is the DNA. So this is the structures of the DNA. From this DNA, as we know that the formations of the messenger RNA formations from the DNA, and that process is actually called the transcription. Transcription is the process through which the synthesis of the messenger RNA from the DNA. Okay, from the messenger RNA, you will see the synthesis of the different proteins. Okay, now the synthesis of the proteins from the messenger RNA. And now this process is actually called the translation. Okay, synthesis of the proteins from the messenger RNA. Okay, after that you will see the synthesis of the proteins. After the translation, you will see some modification in the protein, and that why this is process is actually called the post translational modification in the protein. Okay, that is the post translational modification. Okay, modification in the protein, that is actually post after translation process. Okay, so most important modification actually happens in the presence of the vitamin K is the gamma carboxylation. Okay, specifically if you see, as we know that the proteins actually made up of different amino acids. So you can see easily here are basically the different molecules of the amino acid. So most important amino acid is the glutamic acid. Glutamic acid is the most important amino acid in which you will see the gamma carboxylation in the presence of the vitamin K. So that's the most important thing. In the proteins, you will see the gamma carboxylation specifically at the glutamic acid amino acid. That's the most important thing. Okay, but the question is which proteins show the gamma carboxylation. So we categorize different proteins into two: clotting proteins and the non-clotting protein. Clotting proteins, or you can say clotting factors, that are helpful in the formations of the clot. Basically, you will see in the hemostasis, you will see the primary hemostasis, you will see the aggregations of the different platelets. After that, you will see the formations of the mesh around the platelets with the help of the fibrin. So that's the most important thing. What clotting factor actually present? So you will see clotting factor 2, clotting factor 7. Okay, and it is equal to the 9, and 9 is not is equal to the 10. So here actually the mnemonic, you can remember it, 2 plus 7 is equal to 9, and 9 is not is equal to the 10. Here is the mnemonic to remember the different clotting factor, vitamin K dependent clotting factors. So vitamin clotting factor 2, 7, 9 and the 10. Okay, non-clotting factors, or you can say proteins, you will see most important is the osteocalcin, osteocalcin, matrix proteins. Actually they are actually present in the bone, matrix proteins, and you can see the protein S and the C. Protein S and C actually break the formations of the clot. Okay, here are basically the different proteins in which you will see the gamma carboxylation with the help of the vitamin K. So we are going to discuss gamma carboxylation. So as the name indicate, gamma, what does it mean? So here, if you see here, here is the structures of the glutamic acid. So basically amino acids contain the 4 major group. That if you see here, center will be the carbon. At one side you will see the presence of the hydrogen group. Okay, at the other side you will see the carboxylate group. Okay, on the other side you will see the presence of the amino group, and that is the alkyl group. Okay, that is actually the glutamic acid. This is actually glutamic acid molecule. Okay, but the most important thing, in our body, glutamic acid is not present in the neutral form. Basically, in our body, glutamic acid present in ion forms, and that is actually called the glutamate. Okay. So if you see here, here is the carboxylate group and remove of the H, and you will see the presence of the negative charge. Okay. Similarly, from here, you will see the remove of the H and presence of the negative charge. Now, this ion form of the glutamic acid is called the glutamate. Okay, that is actually the glutamate. I write it here, glutamate, and you can write GLU. That is the glutamate. Okay. Similarly, you can see this is the one amino acid. Other amino acid will continue both sides, at the carboxylate side and at the amino sides. Okay, but in that situation you will see one hydrogen will be less as compared to the amino group. Okay, here is basically the glutamate ionic form. Okay, it, you will see just additions of the carbon dioxide and formations of the carboxylate group. That is actually the carboxylation. So here is the our main functional group. Beside the functional group you will see presence of the alpha group, alpha. After that you will see the beta carbon, and third one is the gamma. Okay, here is the main functional group, alpha, beta and gamma. Now you will see the additions of the carboxylate group at the gamma carbon. So that's why we call this process gamma. Carboxylations mean you will see the additions of the carbon dioxide and formations of the carboxylate group. So if you see here, we can remove the H because it is actually present in the ion form. Similarly, at that side. Okay, you will see this is the additions of the or formations of the carboxylate group. Okay, but the question is, where does this carboxylate group comes from? So actually here is the process. In this process, the most important enzyme here is the cute enzyme, and that is the gamma glutamyl carboxylase. That is our most important enzyme in this process, gamma carboxylation. Okay, in the enzyme you will see the two sides. First is the active side, and second is this side you will see the attachment of the cofactor, or you can say coenzyme. Okay, at that side you will see the attachment of this glutamate residue attached to the active side of this enzyme. Okay, and second is for the coenzyme, or you can say cofactor, and our main cofactor or coenzyme in this reaction will be vitamin K. Okay, so actually, and you also see the additions of the carbon dioxide and the oxygen in this group. Okay, and after that you will see the release of the water in that scenario. Okay, so question is, where does actually vitamin comes from? So actually vitamins present comes from the intestinal microorganism and through the diet. So actually vitamin exists in quinone form, manaquinone, phyloquinone. So actually here is the structures of the quinone, general structures of the quinone, which we have discussed in the previous lecture. You can check it. Okay, so basically this is the quinone form. It will first convert into the hydroquinone form of the vitamin. Okay, that's the most important thing. Here is the structure of the hydroquinone. That is actually the reduced form, reduced form of the vitamin K. Okay, that is the quinone form. It will convert into the hydroquinone with the help of the enzyme, that is the vitamin K reductase. The enzyme is the vitamin K reductase enzyme. That is the most important enzyme. Okay, so what the difference between the quinone and the hydroquinone? You will see presence of the hydroxyl group at that site, but in the quinone you will see the oxygen group. Here is the difference. It means that you see the additions of the two hydrogen atom, and that is actually the reduced form. So in this reaction you will also see that where does these two hydrogen atoms comes from. So basically along this reaction you will see NADH2 convert into the NAD positive. Okay, now in that reaction two hydrogen atom is given to this molecule quinone and it will convert into the hydroquinone. But this reaction will happen in the presence of the vitamin K reductase, means that this enzyme will do reductions of the quinone and formations of the hydroquinone. Okay, that's the most important, and this is the active form. Hydroquinone is reduced and the active form. Now this form of the vitamin will attach to the enzyme, and that is gamma glutamyl carboxylase enzyme. Okay, now our things has been ready, that is the cofactor, or you can say coenzyme. Second is the substrate, our glutamate residues. Okay, and in that process you will see additions of the carbon dioxide, additions of the oxygen. Okay, after the reaction you will see the remove of the water molecule, that is our second focus, and the ultimate product is this one, that is the additions of the carboxylate group. Okay, now this after the reaction this cofactor, that is the hydroquinone, will convert into another form of the vitamin, that is the epoxide form of the vitamin K, and in that scenario you will see here is the oxygen. Now you will see the difference between these two, hydroquinone and epoxide form. You will see oxygen, and at that side you will see the oxygen instead of the hydroxyl group. Now this is actually the oxidized form. This is actually the oxidized form. Why do we call it oxidized form? Because at that side the hydrogen atoms has been removed. So ultimately simply is that carbon dioxide convert into the CO, C double O negative. Okay, and this is oxygen. One oxygen comes to this form and one oxygen comes to the water, and two hydrogen move to the water molecule. So that is our main process. Ultimately you will see the formations of the epoxide form. Now this epoxide form again convert back into the quinone. Okay, with the help of an again another enzyme, and that is the vitamin K epoxide reductase enzyme. Okay, that is the most important thing. Again in this process you will see the additions of the two hydrogen atom and the removal of the oxygen. So in that scenario, you will see thiol, dithiol, you will see the dithiol convert into the disulfide form. Okay. In this process, here is a compound, and this compound actually gives the two hydrogen. Now, these two hydrogens carry the oxygen and ultimately remove of the water and conversion of the epoxide to the quinone form. Now the oxygen atom has been removed and that in the form of the water, and now this is the quinone form, and again this will convert into the hydroquinone form, that is the active form, and again can be utilized with the gamma glutamyl carboxylate enzyme. So here is basically the three most important enzymes that are helpful in the gamma carboxylation, and now this whole process will happen in the rough endoplasmic reticulum. Okay, and last most important, these enzyme vitamin K epoxide reductase and the vitamin K reductase actually inhabited by the warfarin, and ultimately you will see this carboxylation stop. It's mean that formations of the clotting factor, active form of the clotting factor will reduce and ultimately clot will not form. Okay, so this is all about the sources of the vitamin K and the different process of the or different functions of the vitamin K. If you still have any question, you may ask in the comment section. Thank you so much.